which is effect-size.ipynb. Read through that, execute the code, see what it does, and when you get to the first exercise, do some work on that. When you get to the part stop there. One other suggestion, which is consider working with a friend. So if you haven't done pair programming before, this is an excellent chance to try it out. Introduce yourself to a neighbor. A couple of reasons that this, I think, will help. One, you will have twice as much screen area. So one of you can have the slides or documentation up, and the other one can have the notebook and execute the code. The other is you've got two people to think at two different levels. So one of you can be thinking about understanding what's going on and thinking about what you should be doing, and the other one can be thinking about the details of Python and Jupyter and how to do what you should be doing. So take a minute. I dare you to introduce yourself to your neighbor. If you want to do it without cheating and no looking, I will tip you off that almost everything can be done by copying and pasting from the examples above. So the intent of the exercises is really just to engage your brain into digesting some of the details and then putting them into use. If you find yourself stuck on something in an unproductive way, if you're working on it and just not making progress, one of the ways that you can cheat is for each notebook, like effect size, there's another notebook called effectsizesolution.ipynb. And if you open that up, you will discover that it has solutions to the exercise. So do whatever works best for you. Cognitively decide whether you want to engage with the exercises, or you could just run the solutions or copy from the solutions into the other notebook and play around with it. Have people, raise your hand if you are close or nearly done, done or nearly done with the first exercise. Okay, so let me take a minute to just debrief on what we've seen so far. So one of the things here in passing is the random variables from scipy.stats. Have people seen random variables before from scipy.stats? Okay, so this is a newish thing to many of you. And the standard deviation of male and female heights, so we can use that to generate random values like these samples that we're working with. And the other thing that is there is evaluation ECF, which is useful for many stats. So here we're evaluating the ECF density function of the Gaussian distribution. Way to say it. The other possibility there. And depending on the context, those will have slightly different senses. If you have two categories and one of them is sort of like a control, then you might express the magnitude of the treatment relative to the mean of the control. That might be the natural thing to measure relevance. It's symmetric, so there's no obvious right way to put into the denominator there. So arbitrary choice, or you can just put the global mean for both sides. Warm up. Hopefully that's making sense. If you have any questions about it, this would be a good time for them. Anything? So that's just getting from an absolute difference to a relative difference. We'll go back now, but feel free to work with a neighbor going through the next section, and again, when you get to the part that says stop here, stop there. If you are working and making progress, you can feel free to ignore me. Between these two means, there are a couple ways to do that. One of them is to just use the average. In this case, the means is not terrible. An alternative, if they were different sizes, I could do a weighted mean of means. The other possibility is if you look at the two PDFs there, you might choose this point where the two PDFs cross over and choose that as the natural threshold between the two of them. And that's fine too. What I've given you in the notebook is a way of computing that thing. Turns out that that algebraic expression works out to be the point where those two PDFs are equal. I didn't do that analysis here, but if you have a question about it, I can show that to you. In this case, I've got two distributions that have roughly the same standard deviations, so however I choose the threshold, it's roughly going to be the midpoint between them. For this example, it doesn't matter very much. Now, since I have that threshold, we can start talking about ways to express a difference in means in practical terms. So if I tell you that it's 14 centimeters, or I tell you that it's 8%, that doesn't really mean very much. But if you know that you're going to be trying to classify things that are in these two groups, now in this example it's kind of silly. It's sort of like if I tell you someone's height, could you guess whether they're male or female? It's not probably a practical problem. But sometimes the misclassification rate would be useful. Because if you've got two distributions that are wildly different, and practically nobody in between, then you could tell me which group you're in. You could tell me your height, and I would know instantly what category you're in. If there's a lot of overlap, then the misclassification rate would be high. And here's a simple way to just compute the misclassification rate, and it is the number of samples that are above or below that threshold. If you haven't used NumPy before, this particular way of computing things might be new to you. So remember that male sample in this notebook is a NumPy array that has 1,000 heights in it. If I compare it to a threshold, it's going to take each of the values from the array, compare it to the threshold, and it'll either be true or false. So the result of the less than is a logical array that contains 1,000 true or false Boolean values. And now if I take the sum, that seems kind of weird, but it's going to treat all the trues as ones and all the falses as zeros. So the sum is going to be the number of people who are on the wrong side of the threshold. So this says that of 1,000 men, 164 of them would be misclassified if we tried to label them by height, because they fall below the height threshold. If you replace that sum with a mean, try that again. If you make a NumPy call to the method mean, it'll get you the average value of all those trues and falses, which is now in terms of a fraction rather than a number. So either of those is fine, but I'm going to switch back to the way it was before. By the way, Control-Z is a useful thing for undoing changes that you make in a NumPy cell. Okay, so misclassification rate is one way so-called probability of superiority, which in this example is a little bit of an awkward term, but it just means that if we draw a random pair of people, I think of this as the junior high school dance problem. If we choose a random boy and a random girl, what's the probability that the boy is taller than the girl? There are a couple ways to compute this in Python world. Stand aside. Okay. So one way to do this is by lining people up and just pairing them up and computing the fraction of pairs where the female is taller than the male. And you can do that using zip. So if you are more comfortable with Python stuff and you like zip, then you can zip together the male and the female samples, and that's going to pair them up one by one. And for each pair, it'll count the number of times that x is greater than y. Turns out to be about 91%. So if in the US adult population, if you randomly choose a man and a woman, the man will be taller about 91% of the time. And then if you want to get all NumPy with it, you can do the same thing using the logical arrays that I mentioned. Questions? Okay. How many people have you... tell me whether you have got to the end of the notebook or whether you would like a minute to read about Cohen's effect size before I go on. Got to the end? Would like a minute to read about Cohen? Okay, we will take a minute to finish up the notebook, and then I will debrief. Okay, two things I wanted you to see here. One of them is Cohen's effect size, which is a way of quantifying the difference in means between two groups. It's taking the means and standardizing them by dividing through by standard deviations. So it's a way of saying the difference between these two groups is some number of standard deviations. The slightly tricky thing is that it's not clear which standard deviation to use, whether it's one group or the other. And you wouldn't want to compute the standard deviation for the whole population put together, because if it really is two groups with different means, then the global standard deviation would be quite big compared to the standard deviation within the groups. So that's why the denominator there is the pooled standard deviation, which is basically a weighted sum of the two, or weighted mean of the two standard deviations. So the details of that are not super important, except that the denominator is meant to be a measure of the variability within the groups. And this is saying that the difference in means is a certain number of standard deviations. The nice thing about that is that the mean is in units of centimeters in this case. The standard deviation is in units of centimeters. Those two things will always be in the same units, so when you divide them, that ratio, the centimeters cross out, and the result is dimensionless. So Cohen's effect size, one of the things that it's useful for, is making a comparison between different studies. So if I tell you that this study found Cohen's effect size of 2, and that one found that a Cohen's effect size of 1, you can compare those two studies without knowing anything about the domain, without knowing anything about the units that they were measured in. Those things mean the same thing, no matter what. The other reason it's useful is that if I give you Cohen's effect size, you can compute those other two things that I mentioned, the misclassification rate and the probability of superiority. So it's one number that's comparable across different studies. And if I give it to you, you can compute the other kind of important practical things that matter. There's only one drawback to Cohen's effect size, which is that most people don't really know what it is. If you're in a field where it's widely used, you're familiar with this. If not, it takes a little while to get calibrated. So if I tell you that it's 2, For
子熊
example,
みたいな感じ。
that might not mean anything to you until you look at a picture like this. So that's what it
僕は
looks
仲良
like
くないと。
when there's a difference in means of two standard deviations. And you can see from that overlap area, you can get a visual representation of the misclassification rate.
ちょっと逃げましょう。
And if you get interactions working for you in your notebook, which is not a given, but this is the other kind of cool tool that I wanted to show you. If you have not used Jupyter before and you have not seen this, you might like having this slider where you can adjust Cohen's effect size. So when it's 2, that's what it looks like.
これで
And let's see
銃
when
持って
it's
たら
like
殺せるんでしょう
3.
ね。
Ah,
服部さんみたい
interesting.
に。
I'm having the same problem that someone else had, which is... So the way that this is supposed to work is that it updates that figure. But what's happening for me and what might be happening for other people is that instead it's making lots and lots of figures. That's too bad. Well, tell you what, instead of interacting, you might find that it's easier to just use this previous cell, cell number 26, and just plug in different values here. So let's do that. So with Cohen's effect size of 1, that's what it looks like. So that's not a super strong effect. There's a lot of overlap. So the misclassification rate here is half of that overlap. The overlap there is the area, and half of that is misclassification. The probability of superiority is now down to 74%. If you are into psychology, differences between men and women in psychology tend to be like 0.5 or less, and that's what that looks like. So when you see newspaper headlines that talk about all these differences between men and women in various cognitive ways, try to find the effect size, and if it's 0.5 or less, you probably don't care. On the other hand, if you see something like 3, you should be somewhat impressed. So 3 tells you that basically if you tell me your score on this scale, I can classify you with pretty high accuracy. So 3 scores, 5, meaningful, 0.5, maybe not. That's a great question. So I tell you it's 2. Yeah, so I'm telling you effect size, and I tell you, okay, I estimated that Cohen's effect size is 2. Your natural question is, well, how precise is that estimate? How much might you be off by? And we're going to answer that in the next notebook. Yes, sir. Yes. Yes. So I'm treating everything here as normally distributed. Most of what I'm doing, you could replace the distributions with empirical distributions. So just draw random values from an actual sample rather than from a continuous or analytic distribution, and the whole process would be the same. And in fact, we're going to do some of that in the third notebook. Other questions? Yes, sir. Yes, good question. So I think you're talking about this cell here, where I'm computing Cohen effect size. Since I have a laser pointer, I have to use it. So right here, I'm computing a pooled variance and then a square root. And you're asking, why did I compute the variance and then square root it? It's because I wanted to take the weighted sum of the variances, and I have to do that in variance land. Yes, I might have done some unpacking when I translated this. So here, if you look at the math, I've got S1 squared and S2 squared. Yep, those are the variances. Beautiful. I put it in the 5-point font. Let's see. All right, let's crank that up. There we go. How's that? There. Okay. So to summarize what we've got so far, we have a bunch of different ways to quantify a difference in means. Many of them are useful because they are comparable across different studies. So if I tell you probability of superiority, that is expressed in practical terms, and it is comparable across studies. Cohen's effect size also has this nice property and is commonly used in a lot of fields. So that was part one, which is telling me about effect sizes that are like differences in means. Part two, we're going to talk about differences in proportions, and I'm going to use some real data that came out a few years ago. This is a study of peanut allergies in young people, and in particular it was children, I think they were about 5 years old, who had tested for a certain inclination toward allergies. So it was a little bit of a unique subpopulation. And they actually did a randomized controlled study where some of them were told to avoid peanuts, and some of them were told, yes, you should go ahead and eat peanuts. And then they checked to